Hi everyone, my name is Bongiwe Mahlobo and the title of my talk is DPP4 and FCRL3 define distinct T follicular regulatory cells in human lymph node tissues. So the production of high affinity antibodies is highly dependent on interactions between TFH and B cells within the germinal centers. However, recent studies have identified a novel subset of CD4 T cells known as the TFR cells, which also localize within the germinal centers. Now, the precise functions of these subsets are yet to be fully elucidated. However, studies suggest that they inhibit TFH function during HIV infection. These cells also share a lot of phenotypic characteristics. So with the TFH cells, there are also other molecules that are also not unique to TFR cells as they are expressed by other cell types upon activation. So this shows that there are limited phenotypic markers that allow the differentiation of TFRs from the closely related subset of TFH, and this significantly complicates their characterization, and as a result, our understanding of the TFR phenotype as well as the TFR biology is still in its infancy. So this study aimed to identify novel biomarkers that would help us to better distinguish TFRs from TFH using RNA-seq. So we used samples from HIV negative and HIV positive, and then we followed the basic RNA-seq procedure. We then used flow cytometry to validate some of the highly differentially expressed genes between the two subsets. So the first thing that we did was we performed principal component analysis, which allowed us to visualize the transcriptional profiles of the two subsets. So from this analysis, we observed that the cells clustered into two-dimensional spaces, such as that they displayed distinct transcriptional profiles. We then went on and generated a list of differentially expressed genes, and this analysis identified a total of 904 genes, which allowed us to generate a volcano plot highlighting the top 40 TFR upregulated genes and top 40 TFH upregulated genes. So here we observed that both TFH and TFRs, they displayed the expected expression patterns, and most importantly, we also identified novel biomarkers that were not previously reported to be differentially expressed by TFH and TFRs. Those included FCRL3 and JBP5. We then went on and grouped these differentially expressed genes into transcription factors and surface molecules. So although transcription factors are also important in identifying cell types, however, our major focus was surface molecules because unlike transcription factors, they allow the identification of cells for further functional studies. So we selected the top 4 surface molecules for further validation using flow cytometry. So far, we've only validated 2 genes, FCRL3 and DPP4. So in A and B, I'm showing representative flow cytometry plots showing the expression of FCRL3 and DPP4 by TFH and TFRs. And then in C and D, I'm showing summary plots showing the expression of FCRL3 and DPP4. So here we are saying that DPP4 and FCRL3 discriminate TFRs from TFH cells. Lastly, we wanted to determine if these markers are unique to TFR cells. And so far, we've looked at CD8 and Tregs. And from this set of data in A and B, we can see that the expression of DPP4 and FCRL3 on TFRs is significantly higher than on Tregs and CD8. So in conclusion, we have identified novel surface biomarkers for the consistent identification and isolation of TFRs in human lymph node tissues. And our study provides new insights into the biology of TFRs with implication for HIV vaccine design and cure strategies. Thank you so much.